Hello everybody and welcome back to the lecture. So, we have been discussing about the DNA sequencing methods and we have talked about Sanger's method, Sanger's dideoxy method or also known as chain termination method to sequence a whole length of DNA. And we have seen how does it work. It basically uses DNA polymerase and it basically uses the same principle of DNA replication to construct the complementary strand of the target single stranded DNA. And then we have seen that how the fragments can be obtained of different chain length. So, the basically if you have the DNA single stranded DNA 5 prime to 3 prime and this is phosphate and we have used certain ingredient which we call the dideoxy DDNTP and then you get the idea is to stop the replication to stop the constructions of the parent strand at every base pair. So, your chain would be terminated, the synthesis of the complementary strand, this should be 3 prime, this should be 5 prime, should be 3 prime, should be 5 prime. So, your synthesis of the complementary strand would be stopped at every base pairs and that is how you can get the fragments of all lengths of DNA and you can analyze the fragments using the gel electrophoresis. That is what we have seen in the last class. So, today we will take another example shorter one and we will try to see we will see what the fragments are. So, I am taking the DNA from the 3 prime to the 5 prime end. Let us make an arbitrary G C T C A G C T A 1 2 3 4 5 6 7 8 9. Okay. The very shorter one to just to show the another example how the fragments are coming up. So, obviously, so this is your target single stranded DNA for which you want to know the sequence. Now, obviously, such short oligonucleotides or such short DNA cannot be sequenced using the Sanger's method. They are usually longer target DNA that we use for sequencing using the Sanger's method. Minimum they prefer that it has to be longer than 50 nucleobase long or 50 base pairs long should be the target DNA, then only you can proceed for the sequencing. The reason is you have to use a primer and the primer itself is 15 to 20 more long. So, your target sequence has to be a little longer than that. So, I am not drawing the primer part, I will start straight from here. So, first what you have vial, let us take a vial A. Vial A contains DDATP along with all the 4 other DNTPs plus the polymerase. So, now and the DDATP is radioactive labeled. So, what are the fragments that you are going to get in vial A? 5 prime. So, it will stop wherever there is a possibility of picking up A. So, this should be C, this should be G, here is the possibility of picking up A and it will stop there because it does not have the 3 prime hydroxyl group and it will show you the radioactive level. That is the first fragment. Second is C G A G T C G A, it will stop here, 2 fragments. The second is vial, let us say T and that has DD TTP with the labeled one. What are the fragments that you are going to get here? 5 prime C G A G T. So, here you have the possibility of picking up the thymine, and that thymine can be the DDTTP, and here it will stop the chain. Let us see if there is anything else. Yeah, the last one, C G A G T C G A, and the last one is T. So, this will also show you the radioactive level. Third one is vial C, where you have added DDCTP. What are the fragments? Here the C would be picked up, and therefore, it will be stopped. 5 prime C G A, this is your G, then comes T, and here is a C. It will stop there. You can write down along with me also. C G A G T C A T, but this will not have labeled. So, this will be the full length DNA, not labeled. So, this will not be seen in your gel. And vial G, that has DDGTP. Fragments are C G, this C G A G, another here. Is there anything else? No other possibility. 1 C, 2 C, 3 C. So, there should be 3 G A G. So, C G, there 1 2 3 C. So, there should be 3 G. First one is C G, 1 2 3 4th position, 1 2 3 4 position, and then there would be another C G A G T C. Sorry, made a mistake. This is C, this is G. So, opposite is A, then this is G, then opposite is T, then G opposite should be C, then should be a G here. So, I need to recap a little bit. C G A, fine, that will stop here wherever there is A. So, opposite to T, 1 2 3 4 5 6 7 8, 1 2 3 4 5 6 7 8, fine. Here vial T, T would be 1 and 2, 2 T's 1, and there are 2 T's. C, now C, there are how many G's? 1 and 2 G's. So, there will be 1, the first one, 1 2 3 4 5 6th position, 1 2 3 4 5 6th position, that is fine. And here there will be this, one is in the 2nd position, another is the 4th position, another would be 1 2 3 4 5 6 7th position. Now, you have the fragments. Now, if you want to draw the gel, let us say this is your vial A, this is your vial T, this is your vial C, and here is your G. Now, let us count the fragment. We have total 1 2 3 4 5 6 7 8 9, 1 2 3 4, 5, 6, 7, 8 and 9. So, first length is here in the vial C, vial A 3, 1, 2, 3, 4, 5, 6, 7, 8, 2 spots there. Vial T 1, 2, 3, 4, 5, one band at 5, and then 1, 2, 3, 4, 5, 6, 7, 8, 9, not the last one. And then vial C, the first 1, 2, 3, 4, 5, 6, you should have one at 6, and then the vial G 1, 2, 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7. So, at the length of 7. Now, do we have all the lengths? This is first, second, third, fourth, fifth, sixth, seventh, eighth and ninth. Now, you can write down the complementary strand of which you are synthesizing from the 5 prime end. So, this would be your C, next is would be your G, third one is your A, fourth one G, fifth is T, sixth is C, seventh is G, 8 is A and 9 is T. So, your complementary strand sequence is 5 prime C, G, A, G, T, C, G, A, T. Therefore, the original strand, the target should be your 3 prime to G, C, T, C, A, G, C, T, A. So, it matches with the original strand. So, that is how obviously you can write the fragments and make the gel and then read the gel accordingly. All right. So, now we have learned how to analyze the gel electrophoresis or the autoradiogram and get the sequence of the your target DNA using the Sanger's method. If we just give you the diagram of the gel, you can find out what is the original DNA, and of course, the vice versa. If I give you the original DNA, then you can find the gel out also. So, now let us move on to the other sequencing method, that is the Maxam Gilbert DNA sequencing. Maxam Gilbert DNA sequencing method. This is purely a chemical method. Which means we do not use any enzyme, no enzyme is involved, it is only chemistry. And so, how is it done? It is done, the idea is to cleave the DNA strand in its backbone. So, you cleave the target DNA, strand in a controlled way. So, if you have the target DNA, using chemical treatment you actually break the strand, obviously in a controlled fashion, so that you can have restrictions to certain quantities, that some groups would react under that condition, some other nucleobases will not react under that condition. We will see it shortly. So, since you cleave the DNA target, obviously it is a destructive process. During Sanger's method, you have seen that you had your target DNA, all right. What you have been doing, you have been trying to synthesize the complementary strand, which means your target DNA was always untouched and it could be reused again and again in the cycles. So, it is a non-destructive process. On the other hand, Maxam Gilbert method is a destructive process. You cannot recover your sample, because your sample is getting cleaved or getting destroyed under the treatment of the by the chemicals. It is a destructive process and it is a selective cleavage of the purines and the pyrimidines. We will see there will be some chemical treatment done which will only react with the purine nucleobases like adenine and guanine, and other chemical treatment will act only on the pyrimidines, which means thymine and cytosines. So, there will be total 4 different kinds of reactions, 2 set for the purines and other 2 set of reactions for the pyrimidines. So, 4 different types of chemical treatments. That is what would be done in this method. So, let us take a target DNA. I will write the sequence later. So, this is your, again we use the single stranded DNA, and this is again divided into 4 vials, vial 1, 2, 3 and 4. So, basically all the vials contain a little bit of your target DNA. Now, each of these vials are subjected to different chemical treatments. Are treated with a different set of chemicals. So, let us take one vial at a time and we will see what are the chemical treatments that are done and what is the outcome of it. How does it cleave the DNA in a specific manner. So, vial 1. So, before that there is another step that we have to do to start the reactions. So, let us take a sequence. Now, the P and this is the 5' end, I am writing now the sugar, S for sugar. Your skeleton has a sugar C GCTA C G T A 3 prime end. So, let us take that this is your target DNA, the sugar, the base, sugar base. And again now the same thing as Sanger's, that you need to track your molecule, whatever the fragmentations you are doing, whatever the chemical treatments are doing, it is going to yield some products, and you need to identify the products, or you need to track the products, where are they located. So, we also will need a labeling here as well, and that is the first step that is being done, that selective labeling of this 5 prime phosphate by P32. This is usually done by enzyme. So, what you get your actual target P32 5 prime G A C G T A. So, now you can track wherever it breaks. Now, vial 1. So, first treatment is done with a chemical reagent that is known as dimethyl sulfate. Dimethyl sulfate. I will show you the structure. This is sulfur, OCH3, OCH3. This is called dimethyl sulfate. I am giving an abbreviated name DMS. So, this compound is usually used as a methylating agent. Methylating agent means if you want to methylate something, then this is used, so that one methyl from here would be transferred from here to your compound. The reason is, if you have a suitable nucleophile, so because of this internal resonance, this is very labile, so you can easily pick it up. So, methylation can be done, and this methylation is quite selective. It does not methylate all the nucleobases. So, primarily the nucleobases would be methylated by the DMS, but it is a selective methylation. It does not methylate all the nucleobases. It methylates guanine, at N7 position. We will see the structure, and it methylates adenine at its N3 position. So, reaction centers are different. Do not methylate, or it does not methylate thymine and cytosine. So, it is a selective methylation only for the purine nucleobases. Now, if it methylates, then what happens? So, if you look at the DNA helix, there is major group and minor group, and if you look at the base pairings, I will look at the CG base pairing first. Here is your sugar, and here would be the carbonyl. This is the 5-member ring. Here is your sugar. NH double bond N, correct. Here is your sugar, and this is your N7. This is N7. So, this nitrogen would be methylated by the methyl, and this will be plus. Now, if you look at the helical form, so the interior part of the sugar, this is the sugar moiety, this is the interior part of the helix, and here this part actually constitutes a minor group of the DNA, and this part, the upper part, is exposed to the major group of the DNA. So, which essentially means that your methylated version, your methylated guanine, this is your G and this is your C, is exposed towards the major group of the DNA. Now, let us look at the AT. Sugar, here would be N. This is your A and this is your T. A is this. This is your sugar, here is the double bond, yeah. So, this is your A, this is your T, and for adenine this is the N3 position. So, here it is methylated, so N3 of adenine, and now again this is the interior part, this forms the minor group. The upper one is the major group. So, you see here the methylated version of the thymine is exposed to the minor group of the DNA, and that actually makes a quite difference, because so once you treat this with dimethyl sulfate with DMS, then you get these things with the sugar. Now, followed by a treatment with hot piperidine. So, if you write down, this is the 3 prime, I have to go to the next page I think, and here your G with the methylation version will be not star, I will write Me, and we will see that actually what happens is when you treat it with dimethyl sulfoxide and then followed by treatment with piperidine, then only the G which is there, that would be cleaved off the sugar. adenine will remain intact, because the methylation happens in the minor group kind of. So, if I write down the actual structure, here is your sugar. I have drawn it in opposite direction, that is not correct, it has to be the other way around. So, this is DNA, this is DNA, and here is your N, here is the methylated version. So, it is plus, and in here you will have the amine NHCO. This is your guanine. Now, what happens is, I will shortly show you brief, I will not go into the details. So, once this have the methylation, this will be picked up. So, that creates a positive charge here, and it will be attacked by water molecule, and what you are going to get is there will be water, NCH3, here is your sugar, and here you have this. Now, this forms a ketone and it actually opens the ring up, this breaks this up. And you get, then ultimately it chops it up, it will open up as a, let us write NCH3, this should be NH, and it would be the sugar, and here it would be CO. So, your nucleobase is cleaved, and now under the condition of piperidine treatment, this will be cleaved up, water will come here. So, basically what you get is this temporarily, your phosphate and the DNA. So, you get the sugar without the nucleobase, and this is called abasic sugar. Abasic means sugar and no base. Sugar without the base is called the abasic site. Once you get the abasic site, then in piperidine what happens, it gets oxidized, and that will basically open up, it will make the cleavage of the 5' end. So, different people show different kinds of mechanism. So, it is something like this. So, you have OH here that has cleaved out, and you will have the ketone, the aldehyde here. The aldehyde always reacts with the piperidine. This is the structure of piperidine, and you get, so this will be ultimately be chopped off. So, effectively you get this, it is not OH, this is, and there is C, it may be in terms of O or it may be in terms of that other different thing, and this prompts cleavage of this also. So, ideally what you get is by the treatment of DMS plus piperidine, you get a cleavage. If you have the G, then you get a cleavage here, that prompts you to get a cleavage here and a cleavage there. So, both the 5 prime end of the DNA as well as the 3 prime end of the DNA is cleaved off. What does that mean? So, if you have a DNA G and if you treat it with the DMS plus piperidine, what essentially it does, it will cleave this off. You will get a fragment this plus a fragment this. Now, we have started with the P32 labeling here. So, therefore, this fragment will have the labeled one, this fragment does not have the label and we will not be able to visualize it. So, we do not consider this strand, we will only consider the fragments which will have the labeled phosphate. And we will see in the all the subsequent steps, it basically does the same thing. It chops off the nucleotide from the 5 prime end as well as the 3 prime end. So, that is the first treatment. Forget about this now. So, we had the sequence, please do not consider this one for the time being, which was 5 prime phosphate and we had the sequence which was G. Now I do not have to write this because we have already seen how this is cleaved off. Phosphate, then you have GC TA CG TA. Now, so vial 1, what kind of vial 1? We now call it a G, because vial 1 will cleave only the G, not the A. So, fragments. So, first one is G, G would be chopped off here, it is gone. So, you are left with only the phosphate. Next P, I have to write the phosphate all the time because this is one band that will that you are also getting. Phosphate G C T A C C, because G would be chopped off here and that is all. So, this is about the fragments that we will get in vial 1. Now, vial 2, the treatment is same dimethyl sulfate plus piperidine. So, it will basically methylate guanine and adenine. Now, if you do this treatment in presence of formic acid, so that brings the pH low, and in formic acid your glycosidic bond means this bond, basically this bond is called glycosidic bond. So, this bond after the methylation gets very weaker and therefore, this cleaves both G and A. So, wherever you have the methylation, wherever you have a G, wherever you have a, both will be cleaved off under this condition. So, vial 2 we call it now G plus A, it will cleave G, it will cleave A both. So, what are the fragments you will get? You will get this all fragments that you get from G plus the A. Second is P, G, C, T, here you will stop at T. Is there anything else? P, G, C, T, A, C. G would be chopped off, G, C, T, A, C, G and T, A would be cleaved. So, we will get these fragments from the vial 2, which basically destroys both G and A. So, this is also a kind of double check method, where you can get two bands and can verify with the other. So, that is vial 1 and 2. Now, purines are done. Now, vial 3 will treat it not with the DMS or not with piperidine will be there. We will treat this your target DNA with hydrazine. Hydrazine is NH2 plus piperidine. So, what this will do is hydrazine will react with the pyrimidine nucleobases. So, it reacts with both thymine and cytosine. I will just with one example I will show. If it is thymine, this is sugar, this NH, this is your thymine. If we treat it with hydrazine, so if you treat hydrazine with a carbonyl compound, it will give you hydrazones. There will be condensation reactions here or here. I will take the example of this. So, here it will form this kind of condensed product. Now, once this is formed, this undergoes intramolecular rearrangement and basically breaks this up. So, under piperidine treatment, you basically get again the same A basic sugar. Followed by again the breakage of the 3 prime 5 prime end as well as the 3 prime end. So, this treatment hydrazine with piperidine cleaves both thymine and cytosine. So, if your sequence I have to read again. GCTA, CGTA, that is the prime sequence. So, the fragments in vial 3 would be here parallelly P, wherever there is T and C, G, it will be cleaved here and it will stop. You will have the, so this is 1, P, G, C, it will cleave the T also, done. G, C, T, A, another C is coming, it will stop here. G, C, T, A, C, G, T is coming next. So, it will stop there and that basically it, you will not have any other. The last one, the full length C, G, T and A, the full length. So, these are the fragments that you are going to get from vial 3. So, vial 3 we now call it T plus C or C plus T. And next now the third last one, vial 4. Vial 4 is treated with the same hydrazine plus piperidine in presence of 2 molar sodium chloride. Now, if you do this in presence of sodium chloride, then what happens? This does not, your thymine does not react here. Under the presence of sodium chloride, thymine does not get cleaved. So, all you see is the cleavage of only cytosine. So, wherever there is cytosine, it will be cleaved, not the thymine. So, again I have to write the target GCTA CGTA. What are the fragments? If it cleaves only the C, G, P starts with P. Here is a phosphate. It is important actually, because this we have to count. P, G, C, T, A, C would be cleaved. G, C, T, A, C, G, T, A, the full length. So, again if you compare with vial 3, you will get all the fragments that are there in vial 3, and vial 3 will contain some more fragments for thymine. Now, you are basically done. So, you again have, like the Sanger's method, you have the fragmented DNA with all the lengths. Now, if you try to construct the, do I have space? I have to do it here. The gel, your gel will be, so G, next one is G plus A, next one would be your C plus T, and the last one would be your C. And here are the length starting with the first phosphate. So, we will count the phosphate also. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now, you can actually do it. The first length will be the single phosphate that we have got, single phosphate here, that was obtained from vial G. So, G vial, first vial, G vial contains the 1 and 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 1 and 6, 1 and 6, 2 bands here. Now, G plus A, 1, 1, 2, 3, 4, 1, 4, 1, 2, 3, 4, 5, 6, 1, 4, 6, 1, 2, 3, 4, 5, 6, 7, 8, 1, 4, 6, 8, 1, 4, 6 and 8. So, you see you can match G and G plus A, or whether your gel has whether fragmentations has occurred or not. It is a double check up actually. So, vial 3 is C plus T. What you have is 1, 2, 1, 2, 3, 2, 3, 2, you have 3, 2, 3, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 7 and 9. So, 5, 7 and 9 I guess. 5, 7 and 9, and lastly the C has 1, 2, 2 is here, 2, 1, 2, 3, 4, 5, 5, 1, 2, 3, 4, 5, 6, 7, 8, 9, and the last one is 9. And you can read your sequence now. So, this would be 5 prime to, if I write, this is G, G means G plus A also you can match. So, this would be your G. Next is second length is C plus T. So, and it is C is there. So, it is C. Third length is C plus T, but it does not show up in C. So, this has to be T. Fourth is G plus A, but there is nothing in G. So, this is A. Fifth is C plus T, and it is present in C. So, this has to be C. 1, 2, 3, 4, 5. So, with the phosphate, so this is 6. Next is this. This is G and G plus A. So, this has to be a G. Next is this C plus T, but there is nothing in C. So, it has to be a T. Number 8 is G plus A, but there is nothing in G. So, this has to be an A, and the last one is C plus T and C. So, it is C, G plus A, fine. Then C plus 2, C, G, C, T, A. So far is good. C, G is also correct. C, G, 1, 2, 3, 4, 5, 6, 7. 7 is this. 7 is C plus T. So, this is A, this is T, and the last one, this has been a mistake, I think. This has been a mistake somewhere. It has to come here. It has to come here. A. It has to come here. So, that would finish it. 1, 2, 3, 4. G, C, T, A, C, G, T, A. Yeah, that will be it. Okay, so from the gel you can read the strand, and that will be your original strand. So, these are the fragments. It is a little bit complicated process, but very fine chemistry actually, and very controlled chemistry that are quite selective to certain nucleobases and will not disrupt the other nucleobases. So, primarily this is a destruction of the DNA and destruction of the main DNA strand. So, with this I will complete the module 3. So, what we have covered is that we have seen the process of DNA replication in the living cells, how the polymerases act, and how the other enzymes functions also or take part in the process of DNA replication. Second, we have seen the polymerase chain reaction PCR, how you can do the replication, same replication process, or you can amplify the quantity of your DNA in a laboratory using the chemical technique, that is called the PCR in vitro. So, third, then real time PCR, how can you monitor the PCR that is going on, how can you monitor whether you are getting your correct DNA amplified or not using the fluorescence tags or using the fluorescence markers. And then DNA sequencing, we have covered the Sanger's method, which is dideoxy method or also known as chain termination method. And finally, today we have covered the Maxam Gilbert method for the DNA sequencing. Thank you.